In the last part of this chapter, we're going to talk about gene regulation and the translational and post-translational level. So when we're talking about gene regulation at the level of translational control, what we're talking about is the degree to which the mRNA is translated or turned into its protein product. So some of the factors that depend on this, or the features of the mRNA, are going to affect whether or not, first of all, whether or not translation occurs, and then secondly, how long that mRNA remains active. So things like the presence of the 5' cap are going to affect whether or not the ribosome is going to be able to find this particular mRNA and is going to be able to start translation. Things like the poly A tail on the 3' end are going to affect the stability of this mRNA. And if it's lost, that mRNA is going to be turned over and it never can be turned into a protein because it's not going to be able to be translated if it's not there. There's a few other ways that translational control can happen. And one of these we'll talk more about later, but microRNAs or miRNAs going to regulate translation by causing destruction of these mRNAs before they can be translated. So you see this sometimes called siRNA or short interfering RNA, shRNA, short hairpin RNAs, microRNAs, and so on. So there's several different ways to describe this. But overall, there's going to be a region of RNA that's double-stranded. And in this region, the cell can detect that double-stranded RNA, realizes that it's a problem because RNA is not normally double-stranded, and it destroys not only that mRNA that looks like it, but every other mRNA in the entire cell that looks like that or has that same sequence as that double-stranded RNA. And so we're going to talk more about it later, but just wanted to at least introduce some of these ideas to you. It can also regulate this effect post-translationally. So once you create this protein, once your cells do translation, finish translation, some proteins are modified after they're synthesized, and we're going to call this post-translational modification. Many times these modifications are crucial to the functioning of that final protein product. This is a relatively newer area of study. Many different types of modifications can be made. These modifications can either enhance or decrease activity of that particular protein. And they can happen in response to many different outcomes or many different environments that that cell might find itself in. When I was a postdoc, there was another girl in the lab who was studying this on the glucocorticoid receptor. The glucocorticoid receptor is just a stress hormone receptor, and it can regulate gene expression. And what she found was that a phosphate group on one particular amino acid completely changed its function in terms of things like insulin resistance for this particular organism. Some other examples, the N-terminus amino acid, a lot of times it's removed, modified. Remember that start one is methionine, so it's not always included in that final protein product. Sometimes individual amino acid residues are modified. Things like phosphate groups can be added to some of them. Carbohydrate side chains can sometimes be attached. Polypeptide chains may be trimmed. You can add or remove signal sequences so that it knows to be targeted to different areas of the cell. And then finally, sometimes these polypeptide chains can be complex with different metals, and this can affect their function as well. So in conclusion this week, focus on answering the Mastering Genetics questions from chapters 13 if you haven't done it yet, as well as chapter 14. Remember, all of these assignments are due at the end of the semester. However, I highly recommend that you don't leave them to the last minute. You might as well get them done as you're going through them. They will also help you with the weekly quizzes. Make sure you're participating in the discussion forums. And this week we do have a quiz that's going to be covering both week 8 as well as week 9. In your spare time, you can also be working on your student project. Next week I will not post any new videos, or if I do, there are not going to be anything due because next week is going to be spring break.